So we often know a lot of the key players, the key genes, the key proteins that are involved in making a metabolic pathway function, because that's building on years, on decades of excellent microbiology research. What we try to now do with cryo-electron tomography is to answer how those proteins are assembled into larger complexes inside the cell. And based on that, we are also able to see how all of those assemblies may be remodeled. So that's particularly interesting when we're thinking about changing environmental conditions for during things like climate change, but it also means that we might be able to identify how different assemblies may be formed when we're thinking about creating synthetic biology solutions. My name is Tomás. I was born in Lisbon in Portugal, and when I was 18 years old, I moved to the UK. I studied biochemistry there for 3 years, and then I did my PhD at the University of Oxford in structural biology. After my PhD, I moved to Marburg, and I've been here for the past 2 years. Marburg is a household name for microbiology. I think for this sort of research is really one of the leading places in Europe. And I came to Marburg because I wanted to do cryo-electron tomography, and I saw a great opportunity and a lot of space for myself to also develop as an independent scientist to apply a method that traditionally hadn't been established here. And there was a space because my current supervisor was very interested in incorporating that into his research. Cryo-electron tomography is a great technique that allows you to span the scales from going from individual protein structures down to the overall cellular context. And the way it works is you freeze your cells, so you preserve the native environment that they were in. Then you need to thin them down so that they are essentially transparent to be seen with the electrons that we use to image them. And then we reconstruct a three-dimensional volume of the interior of the cell. One of the key differences, if you're interested in looking at protein structure, would be that you may preserve certain interactions that you may lose when you break the cells and isolate the proteins. So it gives you all that cellular context, which is really, really valuable. So what we try to do is to describe how the proteins that are performing the functions that we are interested in organize themselves inside the cell, how those whole metabolic pathways are compartmentalized or how they communicate and what else they may be interacting with. So we describe the architecture of the key biological functions that we're interested in directly within the cellular environment. And particularly on the structural biology angle, we have been working with a lot of groups within Marburg, of course, but also broader within Germany. And that's also been very exciting because it's allowed us for us to access a lot of interesting biology and for others to implement the techniques that we use into their research and come up with much more exciting projects than either of us would be able to do. So one of the things that I'm trying to do is that I'm trying to also lower the entry barrier for new users to use the pipelines that we're establishing. And that has the advantage that new students can take up the method within our lab, but also we're trying to set up a hub that can be used by other groups within the M4C to tackle a wide range of biological projects. Since I've been doing that, I've been starting to work more and more with other groups around Marburg and the M4C. So the way I see it, it's been nice that I was able to establish the setup and the techniques to work on the projects that we are interested in, but you can see that other people are interested in, and it's sort of spreading out, which I think has been really great. Marburg is an excellent place for microbiology research with a very long-standing tradition where a lot of very important discoveries have been made. It's a very collaborative environment. It's a place where people get together to tackle something bigger than their own project, and that's what really drives things forward. You can see with initiatives like the M4C how everyone is very keen to develop projects together and to support each other, and that's what I found. So ever since I moved here for my postdoc, very quickly I've started to work with many others. And that support, not only from the project point of view, but also you can see that people value that and they do support your career. That has been a very fundamental reason why I think my time here has been so enjoyable and so successful as well. And my advice to anyone that might be interested in the type of research that anyone does here and wants to join is to just get in touch. I've found everyone to be very open, very supportive. Throughout my time here, I've really found that people are supporting not only your project, but also your career development.